You don't have to smoke to get lung cancer. Here are 5 reasons you might want to pay attention to. We've reached an era where the mere mention of cancer sends chills down our spines, partly because we are living longer. Lung cancer, in particular, is the most notorious outlaw in the global cancer battle. For the longest time, one ironclad rule has been etched into our minds: smoking causes lung cancer. The skull warnings on cigarette packs and the coughing, wheezing smokers in movies and TV shows all reinforce this idea. But here's an unsettling truth that's shattering our long-held beliefs. Globally, about 2.5 million new cases of lung cancer are diagnosed each year. Shockingly, 10 to 25% of those, that's 250,000 to 625,000 people, have never actively lit a single cigarette in their lives. They are called non-smoking lung cancer patients. Now, here's the interesting part. If they weren't smoking, what invisible culprits are silently triggering cancer in their bodies? Is it secondhand smoke, polluted air, or some hidden killer we don't even know about yet? Hey everyone, it's Grandpa here. Today, I'm going to talk about the scientific evidence behind how you can get lung cancer without ever smoking. To crack this case, an international team of research detectives launched a large-scale study code-named Sherlock Lung. They analyzed the tumor genomes of 871 non-smoking lung cancer patients from 28 locations across 4 continents. Their findings were published in the journal Nature on July 2nd. Right from the start, the study revealed something striking. The way lung cancer operates varies dramatically between East and West. When scientists zoomed in on key driver genes, the differences were even more glaring. In European and American patients, CRIS gene mutations were unusually active, appearing 3.8 times more frequently than in East Asian patients. On the other hand, East Asian patients showed a dual dominance of EGFR and TP53 mutations. This aligns with earlier clinical evidence that EGFR-targeted drugs work better in Asian populations. This tells us that lung cancer in non-smokers isn't a single disease. Depending on where you live, your environment, and your genetic background, the root cause could be entirely different. Preventing lung cancer effectively requires tailored approaches. Digging deeper into the genetic profiles of East Asian patients, researchers uncovered an unusual genetic fingerprint, SBS22A. And the suspect behind it was shocking: aristolochic acid. This toxic compound, found in many traditional herbs, is already infamous for causing kidney damage, liver cancer, and bladder cancer. But how does it end up in lung cancer? That's been a mystery until now. The study suggests that certain traditional herbs, or plants containing aristolochic acid, might enter the body through ingestion or inhalation, leaving deadly, irreversible marks on lung cell DNA. When it comes to lung cancer in non-smokers, the usual suspect in people's minds is second-hand smoke. There's no doubt that long-term exposure to smoky environments harms the lungs. But when researchers tried to pin the blame on second-hand smoke at the DNA level, they hit a wall. They compared the genomes of 250 patients exposed to second-hand smoke with 208 who weren't. After adjusting for age and gender, they found only a slight increase in mutations in the exposed group, nothing significant. More importantly, there was no clear link between second-hand smoke and any specific driver mutations or mutational signatures. This evidence points to a surprising conclusion: second-hand smoke might not be as harmful as we thought, at least not in terms of directly causing DNA damage. It's more of a cancer promoter, not the trigger, but a helper pushing already mutated cells toward cancer. Of course, this doesn't mean it's harmless. Let's be real. Smoking around non-smokers in enclosed spaces is just plain rude. With second-hand smoke's direct culpability in doubt, researchers turned their attention to another invisible killer: air pollution, especially PM 2.5. When they compared patients from high-pollution and low-pollution areas, they found that those in polluted regions had significantly shorter telomeres in their lung tumor cells. Worse, high-pollution patients had 30.2% more single-base substitutions, 4 to 5.7% more double-base substitutions, and 19.7% more small insertions and deletions in their DNA. That's terrifying. It means breathing polluted air over time can damage your DNA in ways similar to active smoking. So, moving to cleaner air might be one lifestyle change that helps keep cancer at bay. After investigating external culprits like aristolochic acid, secondhand smoke, and air pollution, researchers discovered the most mysterious mutational signature of all, SBS40A. Its origin remains unknown, but it has a clear preference for EGFR mutations, appearing 3.5 times more often in tumors with these mutations. It's also unusually active in non-smokers. Scientists suspect it might stem from some poorly understood internal biological process. This discovery reshapes our understanding of lung cancer in non-smokers. It shows just how complex this #1 cancer killer really is, and reminds us that cancer risk factors are far more intricate and hidden than we ever imagined. So, what can we do? Live in places with cleaner air, avoid secondhand smoke, and steer clear of aristolochic acid. With these steps, maybe one day, we can truly say goodbye to lung cancer. Make sure to hit that subscribe button, give this video a thumbs up, and share it with your friends and family. Thanks a bunch.